Hello, my name is Sabrina Sinder, and I am a third-year student in the Biotechnology Academy of Spanish River High School. The title of my science fair project is How Exposure to Sounds at Different Frequencies Affects the Olfactory Sensory of the Drosophila melanogaster. My category is HCAD. My abstract is Drosophila melanogaster are incredibly versatile model organisms with many intricate functions, such as the ability to hear and smell. Frequencies have been a popular source of interest for years now, as their unique sound is believed by some to amplify learning and memory retention. In this experiment, the researcher tested the effects of different sound frequencies on the Drosophila melanogaster by testing its olfactory sensory after exposure to different scents in a Y-maze behavioral assay. The researcher used saturated pieces of filter paper, one saturated with a mixture of almond extract and 20% sucrose solution, and the other saturated with vinegar. The researcher gave 3 groups of 10 Drosophila, each 10 minutes each, to explore the Y-maze at different times, playing gamma waves at 60 Hz for the first group, beta waves at 14 Hz for the second, and nothing for the control group. Frequencies were both played at 40 decibels from an acoustic speaker. After exploration, the Drosophila group stayed in the Y-maze for 3 minutes, tested on whether or not they would return to the almond sucrose solution, which was rewarded with sugar. 3 trials for each group were conducted. However, there was no correlation found between the sound frequencies and changes to olfactory sensory. Overall, more research still needs to be done to determine if different frequency wavelengths and frequencies in general have any effect on the animal and or human brains, or if they truly do not have an effect on the animal and or human brains. As sound is so prevalent in life today, it is important to determine its effects on animals and humans. The problem/question is, does exposure to different sound frequencies enhance, impede, or affect in any way the olfactory sensory of the Drosophila melanogaster? The hypothesis is, if fruit flies are exposed to different sound frequencies while going through a behavioral assay testing their olfactory sensory, then their olfactory sensory will be enhanced. The project overview is, in this experiment, the researcher will test the effects of different sound frequencies on the learning and memory retention of the Drosophila melanogaster by testing its olfactory sensory after exposure to different scents in a Y-maze behavioral assay. Sound in general has a tremendous impact on the brain due to how it interacts with certain hemispheres of the brain, such as that which controls smell. The researcher can determine whether sound frequencies in general amplify, impede, or make no difference in the Drosophila olfactory sensory, or whether a specific sound frequency will have a more profound effect on this olfactory sensory. The materials used were a culture of Drosophila melanogaster, 40 microliters of almond extract, 40 microliters of vinegar, 20 microliters of a 20% sucrose solution, P20 micropipette tips, a Y-maze, a speaker for acoustics, a timer/stopwatch device, a P20 micropipette, scissors, a Sharpie, a pencil, and 10 to 15 sheets of filter paper, and alcohol wipes. The procedure is, the researcher first starved a culture of adult wild Drosophila for 18 hours. Once they had been starved, the researcher began the experiment. She saturated a piece of filter paper with a mixture of 40 microliters of almond extract and 20 microliters of a 20% sucrose solution, the reward solution, and placed it at the end of one arm A, of a Y-maze. Then the researcher saturated another piece of filter paper with 40 microliters of vinegar and placed it at the other arm B, of the Y-maze. 3 groups of 10 Drosophila each explored a Y-maze at different times for 10 minutes while either gamma waves at 60 Hz, beta waves at 14 Hz, or nothing was played. Both sound frequencies were played at a volume of 40 decibels from an acoustic speaker. 3 trials lasting 3 minutes each were conducted for each the gamma waves experimental group, beta waves experimental group, and control group to see if a sound frequency in particular would impact whether or not the Drosophila would return to the almond sucrose solution after deducing that it was rewarded in sugar in larger amounts. The data/observations and results are: neither gamma waves at 60 Hz or beta waves at 14 Hz had any effect on the Drosophila melanogaster's olfactory sensory. There was no correlation between the sound frequencies and improved or hindered performance on the olfactory test. The few Drosophila who chose to move from the central chamber did so purely because of having been starving. They were not at all driven by recognizing the almond extract as the rewarded sugary solution. The other Drosophila were completely uninterested in exploring the maze, both without the frequencies and with the frequencies, a result that could have been caused by stress levels, habituation, short trial times, and the like. The conclusion is, The purpose of this experiment was to determine whether or not different frequencies at respective wavelengths would either amplify, impede, or make no difference in the olfactory sensory of the Drosophila. To conduct this experiment, the researcher first starved a culture of adult wild Drosophila for 18 hours. Once they had been starved, the researcher began the experiment, saturating different pieces of filter paper with their respective substances. One was saturated with a mixture of almond extract and a 20% sucrose solution, and the other was saturated with vinegar. The first filter paper piece was placed at the end of one arm A, of a Y maze. The other filter paper piece was placed at the end of another arm B, of the Y maze. The researcher gave 3 groups of 10 Drosophila each 10 minutes each to explore the Y maze at different times, playing gamma waves at 60 Hz for the first group, beta waves at 14 Hz for the second, and nothing for the control group. Both sound frequencies were played at a volume of 40 decibels from an acoustic speaker. 3 trials lasting 3 minutes each were conducted for each group. The study found that neither gamma waves at 60 Hz or beta waves at 14 Hz had any effect on the Drosophila melanogaster's olfactory sensory. There was no correlation between the sound frequencies and improved or hindered performance on the olfactory test. The few Drosophila who chose to move from the central chamber did so because they were starving, not because they recognized the almond extract as the rewarded sugary solution. The other Drosophila were completely uninterested in exploring the maze, both without the frequencies and with the frequencies. Thank you for your time. Goodbye.